Hello, everyone. Welcome to the first lecture of the special lecture series. So I'm Min Zhang from the Department of Statistics at Purdue University. I'm also the Associate Director of Data Science at Purdue University Center for Cancer Research. So this seminar series was originally scheduled as part of the NCI-funded R25 summer workshop on big data training for cancer research. Unfortunately, this year we had to move it online, and hopefully we can still reach our original goal of the workshop. Today we have three panelists. John has helped with setting up the Zoom meeting, handling all the registrations. He will also record the lecture and share the recordings with all of you after the lecture. So now I would like to introduce our webinar moderator, Dr. Nadia Landman. Dr. Landman is a research assistant professor in the Department of Comparative Pathobiology at Purdue University. She also manages the Purdue side of the Collaborative Core for Cancer Bioinformatics, which is a joint bioinformatics core for cancer researchers at Purdue University and Indiana University. She has a background in biochemistry, molecular biology, and computational biology, and received her PhD in 2015 from Purdue. Dr. Landman's research interests include single-cell data analysis and data integration with a focus on immunological diseases of the prostate. With that, I would like to hand it over to Nadia.
Hello, welcome. We're glad you're here. So just a few housekeeping notes. Your microphones will remain muted through the duration of the seminar, but if you have questions you would like to ask Dr. Ratliff, please do so through the Q&A function, which is at the bottom of your Zoom screen. And then after the hour lecture, we will take questions. So thank you again for coming, and now I'll hand it back over to Min.
Thanks, Nadia. It's my great pleasure to introduce our first speaker, Dr. Tim Ratliff, Distinguished Professor of Comparative Pathobiology at Purdue University. Dr. Ratliff is a Robert Wallace Miller Director of Purdue University Center for Cancer Research. He's nationally recognized for his achievements in urologic research. Dr. Ratliff was a member of the team that validated the PSA screening test that is widely used to detect prostate cancer and was a leader in the launch of intravesical BCG therapy for non-muscle invasive bladder cancer, currently the treatment of choice for the disease. Dr. Ratliff founded and launched the Society for Basic Urological Research. His research focuses on immunology and inflammation using prostate and bladder cancer models to address questions regarding activation of anti-tumor responses, characterization of anti-tumor effector mechanisms, impact of inflammation on cancer development, and regulation of anti-tumor immunity. Today, Dr. Ratliff will present on the role of tumor microenvironment in promoting tumor growth. Please join me in welcoming Dr. Ratliff.
Thank you, Dr. Zhang. It's a tremendous pleasure to be here and be able to talk with the group and share with them some information about the tumor microenvironment. So what is the tumor microenvironment? The tumor microenvironment is really an integrated environment that consists of many cell types and extracellular matrix proteins that support the tumor growth. The tumor microenvironment is a highly diverse environment. It is considered pro-tumorigenic. That means it supports tumor growth. But not only does it support tumor growth, it also enhances the ability of these tumor cells to move around and to migrate outside of the confinement of the tumor itself. It's composed of multiple cell types, as I indicated, and these include fibroblasts, and in the tumor microenvironment, they're called cancer-associated fibroblasts, or CAF, endothelial cells, which line the blood vessels and lymphatic vessels These are very important cells that secrete a lot of factors that influence the environment. And extracellular matrix proteins like collagen that kind of form the rigidity of the tumor itself, and a large number of infiltrating immune cells. And something that's probably surprising to most in the audience is the fact that at least 50% of the cells in a tumor mass are not tumor cells, but are other cell types that support the tumor cell growth. Now here's kind of a diagrammatic representation of a tumor microenvironment. And you can see here the large tumor mass itself surrounded by normal epithelial cells and the collagen matrix here. And you see the blood vessels coming in, lymphatic vessels taking materials out, and cells scattered throughout this environment. Sort of a list of these cell types in an abbreviated form are shown below the tumor mass itself. So this highly interactive environment where communication among the various cell types not only influences the phenotype of the cancer itself, those communication avenues also impact the phenotype of the non-tumor cells that have infiltrated this environment. And this whole process together forms a very complicated communication network that supports tumor growth and its spread to other organs. So we have focused on trying to understand this, we and many others, as a matter of fact. This is a major field in tumor biology. But we really want to understand this intercellular and autocrine cellular communication process that leads to expanded tumor growth and the spread of the tumor to other organs. So by doing this, we feel that we can identify really important pathways that may enable us to develop new treatments for cancers of various types. Now here is a much more complicated figure of the tumor microenvironment. And in this one, you get a better idea of some of the structural components. You see surrounding the tumor here, these extracellular matrix proteins and fibroblasts that form sort of a capsule around the tumor. And in some cases, that is a very strong barrier and tumors do not spread. In other cases, fibroblasts and other cells in the microenvironment secrete enzymes that break down this matrix, and they secrete factors that stimulate these little tumor cells in here to begin to migrate. And the combination of the migration and the breakdown of the matrix around the tumor allows these tumor cells to invade surrounding tissue, to invade vasculature and the lymphatics, and to spread to other organs. So at a later time, you can take some time to review this and see these different cell types and some of the functions that they do. I won't go into those in detail right now, but we'll talk about those, some of these aspects as we progress through the presentation. So what I'd like to talk about to the group primarily are the fibroblasts, or cancer-associated fibroblasts, and also immune cells, specifically the myeloid cells and the T lymphocytes. Within the T lymphocytes, primarily CD8 T cells, because they are the primary killers that are activated in immunotherapy to eliminate the cancer. So what we have here are photographs of fibroblasts, normal fibroblasts. These are called 3T3 fibroblasts, and you can see that they're very elongated type cells. They express actin filaments, and this arrow indicates where they're located within the tissues, which is in the interstitial spaces. So they surround the ductal systems and other cellular structures and provide the structural basis for maintaining this organ. So fibroblasts maintain the structural integrity of the connective tissues by continuously secreting these extracellular matrix proteins. And one example, as I mentioned earlier, is collagen. Fibroblasts uniformly express vimentin, and many express actin filaments. Functionally, these cells may be distinct, and we'll talk about that a little more in a few minutes. The fibroblasts secrete growth factors that really drive epithelial proliferation and the development of blood vessels and lymphatic vessels. These cells are very important in wound healing. Wound healing has specific stages, and those stages include a very important aspect where fibroblasts secrete these growth factors, enhance vessel formation, and are extremely important in the restructuring of the wound to a normal tissue process. Part of that process is the deactivation of the fibroblasts that leads to apoptosis of some forms. And this kind of cooling off of the healing process enables the tissue to maintain its normal structure. If for some reason this cooling off part of the wound healing does not occur, what you get in a normal organ is fibrosis. Most of you are familiar with the term liver cirrhosis of the liver. And what this is simply a fibrotic process in which fibroblasts and extracellular matrix proteins are displacing the normal tissue, and therefore the liver loses its normal function. Now, fibroblasts within the tumor itself are called cancer-associated fibroblasts, or CAF. The CAF are a little bit different than the normal fibroblasts. They are present throughout the cancer tissue itself, but really form abundance around the periphery of the tumor, as I indicated to you in the diagram earlier. The CAF function to maintain this structural integrity of the tumor, as the normal fibroblasts do for normal tissue. And again, they produce extracellular matrix proteins, growth factors, tissue remodeling enzymes, and these are the enzymes I was telling you about that break down the matrix and allow escape of the cancer cells. But the point, the real problem for CAF is they are sort of stuck in a tissue rebuilding mode. They don't really cool off. They continue to push for the development and proliferation of the tumors and other surrounding cells, and are really central to the process of supporting tumor growth. They also have the capacity to inhibit anti-tumor immunity. So here's a diagram that just shows you this kind of fibrotic area surrounding this lesion, and that walls it off, of course. But as these degradation enzymes are produced, this area is degraded, and it allows the cancer cells to migrate out into the periphery and seed other organs and form metastatic and often lethal cancers. So how do these cells function? Are they all the same? I mean, they all express certain proteins like vimentin. Do they have separate functions? So that's one of the questions that we have tried to address. And in collaboration with a group in Chicago, we have been working to try to understand what the CAF subsets are doing and if there indeed are various subsets. So Simon Hayward is my collaborator in Chicago and his group, and I'll mention all of them at the end of this presentation. But working with this group in a collaborative sense, we are using single-cell RNA-seq to analyze this transcriptome of cancer-associated fibroblasts. After we obtain the sequencing data, we use unsupervised gene expression patterns to segregate these cells into different subpopulations, if indeed the transcriptomes are distinct. In these initial studies, we performed on primary cell cultures. That means these are freshly isolated calf from tumors that are cultured long enough to expand them so that we can do the work, but not long enough to let them change dramatically. But it enabled us to really evaluate the technology and our ability to segregate these cells into different populations. And again, we're on the road to trying to understand these cells better so that we can develop new therapeutics. Now, here's a heat map that shows that indeed these uniform cancer-associated fibroblasts are not so uniform. They have a distinct transcriptome. They produce different proteins from various overexpressed genes. And Nadia Landman was able to work to segregate these into distinct populations. There are 6 distinct populations in this particular cell line. And this is a SNE plot that shows the separation on an individual cell basis of these cells into their respective subsets. Here we have violin plots looking at 2 very highly expressed chemokines. Now, these are factors that are produced that either activate or recruit leukocytes to the tumor environment. So we focused on 2 of these, CCL2, which recruits macrophages, and CXCL12, which recruits T cells. And in these plots, you can see these arrows at the top, and these arrows indicate whether a particular subset overexpresses or underexpresses at a significant level compared to the mean of all the cells, all the subsets of cells. And you can see that group 0 really overexpresses CCL2, and group 1 really underexpresses, is downregulated in this particular subset. Not significantly modulated in several subsets, but also significantly downregulated in subset 5. Similarly, a CXCL12 is differentially modulated among the different subsets that were identified. And interestingly, CXCL12 is downregulated in subset 0, but upregulated in subset 1, which is the converse of CCL2. So we have very distinct populations that are producing factors that are very important to recruiting the leukocytes into the environment. And here we have a particular type of analysis, Bayesian analysis of gene pathway expression linked to CCL2 and CXCL12. And what you can see is that as pathways that are significantly linked to these 2 factors, we show a consistent and distinct separation of these pathways among the different subsets that we have identified. So just as one example, in cluster 0 that greatly overexpresses CCL2, you see a highly linked pathway analysis system in this particular subset, and not so for CXCL12. which was downregulated in this particular population. Similarly, you can identify pathway analyses that are consistent with the expression of these two genes in the other subsets. So this helps us confirm that what we're seeing is real, and that we need to really better understand these processes. So just to summarize, I don't want to go into too much of the data here, but to summarize, we have identified real subsets of these fibroblasts. Cluster 0 is a very strong producer of CCL2, and it recruits macrophages. And when macrophages are recruited into the environment, other subsets of these macrophages, of these fibroblasts, actually impact other leukocytes in the environment, which produce factors that affect the phenotype of the macrophage, which enhances tumor growth. So you can see from this diagram that it's a very complicated system that single cell analysis is helping us to better understand, and we really hope that in further studies we'll be able to better define very important pathways and alter those pathways. Now let's think about immune cells in the tumor microenvironment. What you have here is the tumor, which secretes some factors like those chemokines and cytokines that recruit all of these different cell types. And of course, when these cell types enter the environment, they likewise do the same. And we have the fibroblasts that are also functioning, so you can see how complicated this whole system is. But all of these different forms of leukocytes have functions within the tumor microenvironment. As you look at these functions, they can be divided into two basic categories that promote tumor progression and factors that promote tumor regression or mediate anti-tumor activity. And you can see that the macrophage can have either of these two phenotypes. The T cell can have either of these two phenotypes, and other cell types within the environment can have them as well. So understanding these critical factors that modulate this whole environment and create either an anti-tumor response on this side, which is desirable, versus a pro-tumorigenic environment on this side, which is not desirable, is very important and will further lead to more effective immunotherapy, which is the tumor regression side. And there's a tremendous emphasis on tumor immunotherapy, and that is part of what we're trying to understand regarding these cells. I'll talk about that a little more in a minute. So as we look at cells within the tumor microenvironment, we see that there are multiple cell types, but I really want to talk about the myeloid cells and the T cells. Myeloid cells are macrophages, which can, as you saw earlier, can be anti-tumor or pro-tumorigenic. And they are important in suppressing the immune system as well as mediating anti-tumor activity. Similarly, myeloid-derived suppressor cells, or MDSCs, can promote tumor growth and can suppress the immune system. We have T cells in the environment, and there are a number of subsets of T cells: CD4 positive helper T cells, CD8 positive cytotoxic T cells, which is what we'll talk about mostly, gamma delta T cells, regulatory T cells, and NK T cells. It's very interesting that these CD8 T cells are present in the environment and are distributed pretty uniformly throughout the environment, but can be turned off by these myeloid cells within the environment. Now, macrophages are very important cells that function not only in tumor immunity in the tumor microenvironment, but are important phagocytic cells in infectious disease, where they can internalize bacteria and degrade them, and internalize almost any particles, and they internalize tumor cells. And this affects their function as well. Now, macrophages are divided into two phenotypes fundamentally. The polar side is an M1 or an anti-tumor phenotype, and the other side is an M2 or a pro-tumor phenotype. The M1s kill the tumor, and that's really what we're looking for. The M2 phenotype supports tumor growth and, of course, tissue repair in a normal organ. They are not killer cells, but rather help the tissue grow and mature. M2 macrophages are the dominant phenotype in the tumor microenvironment. So the environment itself pushes these macrophages as they are recruited to this environment to the M2 phenotype to support tumor growth. It's our job to learn to convert those so that we can generate an anti-tumor response. And of course, they inhibit the immune response and are called tumor-associated macrophages. Now, macrophages come from a common precursor. They're monocytes that are derived from the bone marrow. In the presence of a tumor, factors are sent to the bone marrow that expand this monocyte production. You get more in the peripheral blood. They migrate to the tumor. And as they migrate into the tumor, it's that environment within the tumor that converts them to a specific phenotype. And with certain cytokines, make them M2s, and other cytokines could make them M1s, which is really true in infections, but not so much in tumor immunity. Now then, I want to show you a little movie of the macrophages, and in this case, they're going to be phagocytosing these yeast particles, Candida albicans, and you'll see this one first right here. There it is. It's grabbed by the macrophage, internalized, and now the macrophage will degrade it. That's what an M1 macrophage does. Now this one's going to be internalized, and there we go. It's inside the cell. You can see how this one's being degraded. This one will follow the same fate. So these are highly activated macrophages that are doing their job to kill the tumor or the infectious agent, but that's what we want to do in the tumor environment as well. Let's talk about myeloid-derived suppressor cells a little bit. These are immature myeloid cells that are, again, are derived from the bone marrow, and there are two phenotypes. We won't really talk about that. Some are macrophage-like and others are granulocytic in nature, but they are, the suppressor cells themselves are the immature forms of these cells. And they are migrate into the tumor setting and are modulated by the environment, just like the macrophages themselves. And here's kind of a diagram to show you how that works. They come from the bone marrow and they migrate into the spleen, and they're not really suppressive in this environment, but they can be activated very easily. But when they go into the tumor environment, because of the hypoxia, that's the absence of a sufficient amount of oxygen, what you get is the activation of a genetic code that results in the activation of these myeloid-derived suppressor cells that enables them to suppress T-cell responses. So they turn off those activated killer cells and don't allow them to attack the cancer cells. Similarly, when tumor-associated macrophages migrate in, they are converted to the M2 phenotype. They also have the capacity to regulate these T cells. So you get both phenotypes that inhibit anti-tumor immunity and really effectively shut it down. And that's what checkpoint inhibition immunotherapy that you read about today does. It alters these phenotypes, allows the T cells to expand and then attack the tumor. This is kind of a diagram to show you a little bit of how a myeloid-derived suppressor cell actually regulates T cells. They produce nitric oxide and oxygen radicals, and they modify proteins in the T cell receptor that make it ineffective in recognizing antigens or recognizing the tumor itself. Therefore, they can't kill them. Then there's another factor, a programmed death ligand 1, which binds to a programmed death receptor and blocks that receptor activity. And in doing so, turns off the T cells. So they have multiple ways of actually controlling the T cell response. The T cell receptor here binds to the antigen, CD8 binds, the T cell is activated, and then it will kill the tumor cell. And I've got a little video here that I'll show you of the tumor cells. This, in point of fact, is a virus-infected cell, but you'll see the T cell is bound here. It's reacting with, there it goes, killing these cells. And you see that it's involuting, it induces an apoptosis that is not recognized easily by the immune system and doesn't exacerbate the inflammatory response. So not only do MDSCs use this type mechanism, but TAM also have the capacity to use these inhibitory ligands and oxygen radicals produced by nitric oxide. So let's look a little bit. So this is an area that I study. So we did a gene array in which we took some of those spleens. Remember, the spleen cells for MDSCs are not suppressive, but when they get into the tumor environment, they are suppressive. So we took a non-suppressive versus a suppressive environment and did a gene array and analyzed altered gene expression. There were really over 3,000 genes that were differentially expressed, but one that's really important is the folate receptor beta, which is expressed, was highly expressed on the active cells. So we worked with Philip Lau, began to analyze whether or not folate beta receptor was an important receptor for this. Using an antibody, we could see that indeed the protein was expressed, and we were able to identify the cell population through reverse transcription PCR reaction. We were able to show that the only receptor that is expressed among all of the folate receptors on these myeloid cells is the folate beta receptor. And using technology that's developed by Dr. Lau, we can target, using folate, we can target different compounds to this receptor, which is then internalized by the cell expressing the receptor. And in this case, we're using OTL38, which is a fluorescent compound, so that allows us to deliver this compound specifically to folate receptor expressing cells and identify them. So you can see here that when we deliver these in vivo and then isolate MDSC and TAMs from the tumor setting, you can see that we can identify a very distinct population of cells that express it. Now, interestingly, and as expected, folate beta receptor is not expressed in the spleen, and these cells are not immunosuppressive. So if we take a tumor bearing mouse and we inject this OTL38, we can image that tumor solely by delivering this compound to the MDSC and macrophages that express folate beta receptor within the tumor. So it's a very targetable process that is recognized only by cells within the tumor. There's no fluorescence anywhere else in this animal. And to show the specificity, we inject a very high concentration of folate immediately prior to injecting this fluorescent compound, and you don't see any fluorescence in that setting. So it's a highly specific receptor-associated binding in which we target myeloid cells specifically within the tumor. And you can see here percentages of those cells within the tumor, which is really quite high. And we've analyzed many different tumors, and in all tumors we've analyzed, although the percentages vary, the cells are present in all of these tumors. We look at it histologically. You can see here you have myeloid cells in the tissue. And if we then look at a folate receptor beta, you see that a subset of those cells also express the folate beta receptor. So we can validate the presence of these within the tumor in a very consistent way. We isolate these cells and look at folate beta positive versus negative and their ability to regulate the immune system or to suppress T cell proliferation. And what we see is in a hypoxic environment like that in the tumor, we see that suppression occurs only by cells that express the folate beta receptor for MDSC and for tumor-associated macrophages. So we can identify the suppressive population by identifying cells that express this receptor. And not only can we identify them, we can deliver agents to them. So if you take the same platform that Dr. Lau has developed, and instead of putting an imaging agent on it, you put an immune activating agent on it, toll-like receptor 7 agonist, all this does is stimulate a tremendous cytokine and chemokine storm that activates the immune system and pushes it toward the killer phenotype and away from the non-killer phenotype. So when we do this and target tumors, we are able to induce anti-tumor activity. And while I'm not showing you data, we also convert the tumor environment to an M1 anti-tumor type environment, and we delete myeloid-derived suppressor cells. Importantly, it's all receptor specific because we can compete it off, no anti-tumor activity when we inject a competitor into the tumor-bearing mice. And also importantly, this is a highly toxic compound when delivered without targeting. And you can see immediate weight loss in these animals, and these animals don't do well and require euthanization because of the tremendous toxicity. But that is not true for the TLR7 targeted compound. So we can convert that tumor microenvironment by targeting important immunomodulatory molecules and induce anti-tumor activity and a much more favorable anti-tumor environment. So these FR beta expressing cells are identified, and the receptor is expressed only on myeloid cells within the tumor, and the targeting process really changes their phenotype and induces an anti-tumor response. Now, cell-cell communication within this environment is very important. You can obviously see that because if we deliver this immune activating agent to the folate beta receptor, we change the inflammatory cytokine milieu and we change the process. So understanding what's going on in this process becomes very important. So we want to then do exactly what we did with the fibroblasts and begin to look at subsets of these immune cells to begin to understand what the normal tumor microenvironment is and what a normal microenvironment is, understand how they communicate, and utilizing these, develop new therapeutics. And to do this, we use single-cell RNA-seq again. In this case, we are utilizing an inflammatory process in a benign condition called benign prostatic hyperplasia. This is not a tumor microenvironment, but our work in animal models has shown that inflammatory environment mimics the tumor microenvironment in many ways. And we're able to gain a lot of information by looking strictly at the inflammation within the benign organ, as well as inflammation within the tumor microenvironment itself. So in this case, we are analyzing the inflammatory cells in BPH, and this is a collaboration with the Simon Hayward group at North Shore in Chicago. So here's some BPH tissue. You can see these little dark blue areas are the inflammatory areas. And as we expand this up, you can see all these inflammatory cells within this environment. So what we're doing is digesting these tumors, either small tumors from weakly or non-symptomatic patients, or large BPH specimens from symptomatic patients who have had to undergo surgery to eliminate the symptomatic problems. We isolate the leukocytes from these tissues and then analyze them by single-cell RNA-seq. You can see that as we look at just the leukocyte populations, CD45s, we are able to show basically that inflammation increases in the large tumors. We have 4 large and 10 small that are part of the current analysis. We're continuing to collect large specimens. So we isolate them by flow cytometry, subject them to 10x genomic drop-seq, single-cell RNA-seq, generate unsupervised populations of cells, and then analyze these cells based on known markers within the phenotypes so that we can segregate them, analyze cell-to-cell signaling within these populations. And here's what we have. This is an accumulation of 4 large specimens, 10 small, and we have 3 normal specimens from a colleague at UT Southwestern, Doug Strand. And what we can see almost immediately is, doesn't matter if they're large, small, or normal, the fundamental groups of cells are very similar. As was very surprising to me, especially in this weakly and inflammatory environment in the normal younger men who were organ donors that were utilized to analyze prostate tissue. So what makes these cells different? Well, as we look at the integrated process, here's all the integration. Again, you see very similar. There's one specimen that's a little different. We're not sure what the reason for that is, but nonetheless, it's there. And then you look at the segregated specimen, and again, the overlap, there's this one that is a little bit distinct, but all the others really overlap tremendously. So the cell types within this environment, regardless of the basic phenotype of the environment, symptomatic to non-symptomatic to normal, are fundamentally the same. So we then uncovered, based on known gene expressions for various phenotypes, the types of cells that were segregated in this unsupervised segregation process. And you can see all the same cells that we saw in the tumor microenvironment. But now what we need to know is, well, what is the difference? And to do that, we use some scoring processes that was published recently that allows us to look at autocrine and paracrine communication among cells within the environment. And Nadia has been working to analyze these types of tissues, our signaling processes, and we're trying to understand how these communication processes are functional and where the differences lie among the signaling processes. You can see large numbers of signaling processes that are significantly represented in the different tissue types. And there are differences in signaling among the tissue types. But here's just a diagram that shows the segregation in a UMAP of the different populations that were unsupervised segregation. You can see the different phenotypes of cells within this environment. And these arrows indicate communication from one cell type to another. And the width of the arrow indicates the strength of the communication link. You can see that in this example, mast cells are communicating with a number of different cell types, and some of these cell types are communicating back to the mast cells. Now, we don't understand what this process is right now, but we know that fundamentally the phenotypes are the same among these different conditions. So we want to really begin to appreciate the differences in the signaling so that we can identify appropriate ways to intervene and alter the symptomatic conditions that are developing. Similarly, in the tumor microenvironment, we want to do the same thing, and we want to identify pathways that will enable us to block the development of that M2 phenotype and activate the development of that M1 phenotype that will result in the elimination of the tumor. So, and our work today, a single laboratory just doesn't have the expertise to get everything done. And it requires strong collaborations across multiple disciplines and industry to be sure that you can perform all the types of analysis that you want to. So my laboratory and the people within this lab have been certainly integral, the immunology component, Nadia and Segar in the computational genomics group that are essential to really bioinformatically analyzing these data and presenting them so that we can understand the biology. The Strand lab, who is able to collect normal tissues from organ donors. The Hayward lab, which has expertise in cancer-associated fibroblasts and are helping us really evaluate the inflammatory response as well. The Lau lab, which is a chemistry lab that can develop chemical therapeutics, one of which I showed you, which is a patented compound, that the TLR7 agonist that we hope to move toward clinical application in the near future. And Endocyte was a company formed by Dr. Lau that helped us in many of our studies, and On Target, another company formed by him, have also been integral in helping us move. So this very strong collaborative interaction has enabled us to really begin to focus on developing technology to modulate the inflammatory and tumor microenvironment in a positive way to alter disease progression and hopefully one day eliminate the need for surgery and increase survival in cancer patients. So I thank you very much for your attention, and I'm happy to answer some questions now. So I have quite a few questions already. You've been listening and typing your questions in, and I appreciate that. So how do you segregate or separate CAFs from the rest of the cells in the tumor microenvironment? That's a very good question. That is done through a very mild enzymatic digestion of a tumor specimen, which releases cells within that tumor microenvironment or prostate environment. And we analyze this digestion process looking at various conditions, various time points, and that will enable us to actually isolate an optimal number of highly functional cells. So it takes a little time to get the process down, but once it's down, we can digest them quite readily, isolate the CAFs by flow cytometry. In the case of the data I showed you, we would isolate the cells by letting them grow briefly in culture and then utilizing them in the single cell RNA-seq. We're also doing studies now with direct isolates from digest, and those data are being analyzed as we speak together. Okay, a second question. How do you interpret growth of primary CAF cell lines in culture in the absence of tumor cells and other cells in the tumor microenvironment? Once again, very good question. Anytime you grow cells in an in vitro environment that lacks their normal surrounding tissue counterparts, you alter the cells. So we're aware that these cells are not the same as the cells that would come directly from the tumor. But we chose to do this initially because it was a very short-term culture and the cells retained a number of the characteristic phenotypic characteristics that are associated with cancer-associated fibroblasts. So it was a good test to see if indeed we could do what we wanted to do. And now we've moved to the true freshly isolated cells that we get from the tumor microenvironment. That'll be much more reflective of what is actually going on. Okay, another question. How do you differentiate an M2 macrophage from an MDSC? Well, they have different characteristic markers that we can identify by flow cytometry. We can utilize these different markers. There are mature markers that identify mature macrophages. So we use those to separate macrophages from the immature phenotype, which is the MDSC. So it's really associated with a long history of multiple investigators defining these markers so that we have confidence in the cell phenotype that we are isolating. Okay, we'll keep going with cells, with questions. Is the folate receptor in TAMs and MDSCs of all cancer models, prostate, breast, etc.? How about patient tissues? Yes, the answer is yes. The folate beta receptor is expressed in all the tumor phenotypes that we analyzed. We analyzed lung, prostate, breast, melanoma, multiple prostates actually, and lymphoma. So as far as we know, and can tell in the mouse model system, every tumor has them. Now in the humans, we have been able to evaluate several urologic tumors, kidney and bladder, and both of those have the MDSC and tumor-associated macrophage phenotypes that express FR beta. And we have looked at FR beta in virtually all tumors and reported that histologically, and they're present in almost all tumors as well. Their numbers vary, but they're present in all the tumors. What we haven't been able to do as yet is show function, but we're working to try to do that now. Okay, another question. How does a tumor negotiate the need for extracellular matrix and the need to spread? If you test early versus late tumors, would we see different ECM and stromal gene expression programs? And the answer is yes, you do. As the tumor progresses and develops more mutational events, it alters the environment, it alters the phenotype of the surrounding cells, and can increase the production of these degradation enzymes, which eliminate part of the matrix. And it can alter the type of proteins that are actually produced by the fibroblasts, these extracellular matrix proteins. Some types of collagens, let's just use as an example, are more rigid than other types. And the more rigid types have a different impact on tumor cell expression of genes. So as these cells interact with this rigid structure, it activates gene expression programs that change the tumor phenotype. So it's a very active process that we really have to work hard to try to understand better. Okay, many very good questions here. So how will you evaluate which cell subsets are the most important to tumor growth and metastasis phenotype? Well, we are working hard through bioinformatic analysis to identify important pathways that are associated with these metastatic phenotypes. So as we look at cell-cell communication, we look at macrophages, which are very important. So these tumor-associated macrophages, as I said, are kind of stuck in the tumor growth mode or in the organ cell stimulation mode, and they are producing a number of different factors. And as this environment shifts a little bit, those factors that are produced can vary. And we are hoping to identify specific pathways that are associated with enzymatic profiles that break down the tumor microenvironment extracellular matrix and allow the tumor to escape. If we can shift that phenotype to one that's producing a more rigid collagen type that alters the tumor phenotype in a positive way, meaning it makes it stay in place and shuts down the enzymes that degrade and break down the matrix, then we can keep that tumor confined. So that would be a very positive pathway discovery that we could target and try to activate through delivery of components to the tumor environment itself. So it's not an easy process, but one that we're working hard to try to better understand, and that people like Nadia are trying to develop new ways to help us understand the biology better from the profiles that we are able to analyze on the genetic modulation and gene expression patterns. Okay, another question. What do you think circulating tumor cells playing the role of studying bone marrow samples derived from patients with clinically localized prostate cancer using single-cell RNA-seq, RNA, DNA, and protein techniques to design future trials? Well, that's very important. There's a lot of work going on with these rare circulating tumor cells that actually show that looking at the gene expression pattern of these cells is predictive of aggressive disease versus non-aggressive disease. So isolating these cells and understanding the gene expression patterns of these individual cells also is very important. And you have to understand that, relevant to an earlier question, these cells are now in a different environment. So we not only have to understand the expression profiles of these cells, but we also need to understand what the difference is between these cells and those cells that remain in that tumor microenvironment or are in a metastatic tumor environment. And those may be different. Okay, is the tumor microenvironment and cellular composition of the tumor microenvironment for metastatic tumors usually different at different sites of metastasis or more similar to the primary site? Well, actually, the analyses that have been done evaluating this show that the tumors at multiple sites are different. So there are a number of different studies that really show that the phenotype of the cells themselves are different. They may be derived from different stem cells, tumor-associated progenitor cells within the tumor, and their genetic patterns are quite distinct. The tumor microenvironment, thus, is different, although it remains more of the tumor-promoting environment. And it could be that, and there are some studies that suggest this is true, that the myeloid cells within the tumor microenvironment are really necessary to co-migrate and establish the distant tumor environment so that the metastatic cells can actually grow. So it's really complicated, and that's why we think that by better understanding the primary tumor microenvironment and modulating environment, maybe we can alter that ability of these myeloid cells to migrate out and establish appropriate niches for metastatic development. So we're working on that, as are many other investigators. Okay, have you looked into CAF subsets in the benign versus advanced prostate tumor? How do they compare to the CAFs that you presented in the initial part of your talk? We have not yet done that. We do plan to do it. By studying the benign conditions, we're going to study CAF and other cell types within the BPH tissues, and we will compare those to the same types of cells in the cancer setting. So that's on the agenda. We have a grant that will be reviewed the end of this month that we hope will be funded, that will allow us to begin to look at these different types of cells in the benign setting. And then we're going to work to do that in the cancer setting also. Okay, moving to the next question. Do any other cells other than fibroblasts produce collagen or other extracellular structural elements? And the answer is yes. Macrophages and myeloid-derived suppressor cells produce these factors as well. They are really dominant. The macrophage is a dominant phenotype for producing the same enzymes and for degrading the matrix itself. As I was telling you early in the talk, in wound healing, the macrophage is the cell that really degrades the collagen matrix and allows the restoration of normal cells, proliferation, and reconstitution of the organ. And when those cells don't degrade the matrix, you get this fibrotic condition like cirrhosis of the liver. So macrophages are very important cells in this process too. That's why this whole thing is so complicated, when we have to have so many labs working on it to try to together uncover the true phenotypes and the alterations that can allow us to develop new therapeutics. Okay, next question. Do any other cells other than fibroblast? Oh, I read that one, sorry. Great presentation. Thank you for answering all the questions. Well, I'm happy to do that. Thank you very much for that compliment. I appreciate it. Another question, and any monocyte-derived cells were you able to note in your data? I'm not sure I exactly understand the question, but we did see monocytes, and these are immature myeloid-derived suppressor cells that are really monocytic. But once a monocyte enters the tissue, it's converted to another phenotype very rapidly. And it turns out that the monocytic myeloid-derived suppressor cells mature into tissue macrophages. So that's a process for myeloid-derived suppressor cells, as well as a direct conversion as monocytes enter into tissue macrophages. What were the cutoff size limits utilized for small, normal, and large prostates? 75 centimeters, 70 centimeters, I think. 70 centimeters, cubic centimeters was the cutoff. And that was determined by ultrasound prior to any surgery. All right, I think I've answered all of those questions. Are there any more? Do you have any more questions or any further discussion you would like to pursue? Okay, well, I'm not seeing any more questions come up. I want to thank all of you for your attention. I want to thank you for submitting these very interesting and provocative questions. And it's my hope that you learned something about the tumor microenvironment, the inflammatory response, and our ability to really intervene and develop new therapeutics. So thank you very much. And I will turn it back over to Nadia or Min Zhang.
Thank you so much, Dr. Ratliff. Excellent presentation. And it seems that our participants greatly enjoyed it as well. So thank you so much for taking time to be with us today.
Well, it's certainly my pleasure. And I am thankful that people learn some things, I hope. And I look forward to a great time of learning throughout this whole course.
All right, well, I think that about wraps it up, right, Min?
Yeah, so I would like to thank you again for joining us today. And our next seminar will be